Okay, great. Yeah, so I'm going to talk to you about some tools, synthetic biology tools that can dramatically increase the ability to find small molecule natural products as well as produce them. We just changed our name to Terra Bioworks, so you may or may not find that on the web yet, but we're transitioning from our old name Varigen to Terra Bioworks to better describe what we do. And that is reinvent how we access nature's medicine cabinet of natural product drugs. So biology runs on a DNA operating system, and that's what is used to make the drug. So we produce billion-dollar molecules, and we can accelerate that process by decades if you use the old pharma paradigms, and by multiple years, even with some of the newer paradigms. Traditional pharma companies use conventional methods that can, like I mentioned, can take years to decades to optimize these things. So we're a genome and a metagenome mining company with world-class expertise in big DNA technologies for manipulating complex pathways, including billion-dollar small molecule compounds. And we have IP and revenues, and we're working on our own pipeline of products that we're trying to manufacture. And we've had customers from all over the world, as well as some of the most prestigious universities around the globe. And we've been in business for about 5 years now. So natural products, and I'm not talking about herbal remedies, I'm talking about small molecule compounds from plants and bacteria and fungi, are small molecule therapeutic compounds, and some of them can be extremely complicated and rather difficult and expensive to manufacture, especially semi-synthetic compounds that are chemically modified to produce better versions of the natural drug. So I'm showing two examples here, cyclosporin and plodepsin, 17S-FD895, as two examples of complicated, difficult to make compounds. And using a new generation of tools, we hope to do that much more rapidly and affordably. So in the old days, pharma would use just classical scoop up dirt methods, isolate, ferment bacteria, and look for compounds. And this was really successful up until about 20 or so years ago, when they kept finding the same compounds over and over and over again. And it turns out that in the intervening 20 years, we've had this explosion of next-gen sequencing that has revealed that a lot of microbes have dozens, and fungi have almost 100 biosynthetic gene clusters per genome, and that a lot of them are cryptic. They're not expressed, at least under the conditions that scientists use to find new compounds. So it literally could take a decade to find a compound, and then another decade or two to optimize it with the old paradigm. With some of the newer methods, next-gen sequencing, you can find them much faster, but it still takes a while to push them through to being useful. And what I'd like to tell you about today is using our synthetic biology tools. We can compress that down to a few months using some of our novel genome mining tools to reprogram those pathways. And here's why this is so difficult. The DNA pathway, or operating system for drug production, is very large and complicated, difficult to work with and control. And as you can see, nystatin, it's an antifungal compound, typically used for skin infections. It's 126,000 base pairs with 25 genes. Vancomycin is a antibiotic of last resort, 65,000 base pairs and 34 genes. Not too long ago, there were no good tools to capture and manipulate these pathways so that you can more rapidly access the genes and the proteins to turn them on and manipulate them. And we've cloned both of those pathways. I'll tell you about some more, as well as express them. So the tool that we mostly use for the initial stage of the capture is CRISPR-Cas9. It's the ultimate restriction endonuclease. It has a 21 base pair recognition sequence, so it's very easy with a sequence genome to pick unique sites, flanking biosynthetic pathway, and cut it. And that separates the needle from the haystack. And then we use some other molecular biology tools to capture that pathway and put it into a cloning vector and validate that in E. coli. And we've done this over 110 times now And you can see a lot of antimicrobials and some biocontrol and anti-cancer compounds. So this just shows our scorecard. This is actually kind of outdated. A lot of them are actinobacteria with high GC content, which are very complicated, and some are firmicutes. But more recently, we've been beating up filamentous fungi, which are much larger genomes, 30 to 40 megabases, compared to about 8 to 10 for the actinos. And the cluster sizes have ranged anywhere from 10 to 130 kb, although we've done a couple of them now that are over 150 kb, which isn't on this deck yet. We've also modified these pathways significantly for some, the ones that look promising by deleting things, adding promoters, other refactoring things. I'll tell you a lot more about that. So here's a good example of a customer who had found a pathway from a metagenome, and he had cloned it on 5 different plasmid vectors. The pathway is so large, it spanned more than plasmid could capture. So we took those 5 individual clones and, using overlaps across them, precisely stitched them together seamlessly to develop the full-length BGC from this unculturable marine strain. And that was not easy, given how highly repetitive some parts of this pathway were. And then the next thing that we've also worked on pretty hard is, how do you go about expressing these big pathways? Again, multiple genes, multiple regulatory elements. It's not trivial. So we have come up with matched vector chassis platforms for expressing pathways in Bacillus, Aspergillus, Streptomyces, and a few selected gram negatives. And I'll tell you a little bit more about our approach. We developed this dual inducible promoter plasmid, and basically it uses promoters facing inward to where you would put your biosynthetic gene cluster. And it wasn't obvious that this would work at the beginning, because you're going across so many different genes, pointing in so many different orientations, but it turns out it works great for about 50 to 60% of the time, 70% of the time in the initial cloning phase, to just see what it is and if you can express it. And here's 2 control examples. We used a blue colored antibiotic called actinorodin and a red colored antibiotic called desalprodigicin. Both of these are Streptomyces pathways, and we pulled them out and popped them into the P dual expression vector, and then put them back into a heterologous host, not the same one, but a different Streptomyces. And you can see the native promoters did not work in this context, but the inducible promoters worked from one side or the other and both sides in this context. You get different results with different pathways, but this is a really nice example. And our best example of this process is de-orphaning a pathway for Stravidin with a collaborator at Northwestern University, Neil Keller. They were working on Stravidin. They had the bacteria that produced it, and they had sequenced the genome. They suspected they knew what the pathway was, but correlating the compound, and they knew the structure of the compound, correlating the compound with the pathway was still challenging. And within 2 months of getting the strain from Dr. Keller, we had it pulled out and cloned and sent back to him, and they had run the mass spec on it and shown that it was the authentic expected mass spec size for this metabolite. And they've gone on to show that this works in a publication from last year, I believe, in ACS. And this just shows some more of the details. A lot of times when we're doing this dual inducible all the way across a pathway, we're only seeing 2, 3, 4, sometimes 4-fold increase in the metabolite. But in this particular case, it gave us a 100-fold improvement in the metabolite expression. Obviously, things were lined up really well for that to happen. Here's another example. This is an anti-cancer compound. It was originally discovered to be an antibiotic, but it crosses over into the cancer realm from Lototherapeutics, and it's nargenacin. And here we've cloned this, I forget the size of this thing. It was about 70 kb, I believe, and we're seeing expression even without inducers, but you get a little bit more from one side or the other side, for example. Here's another example of nystatin production from our collaborators at Syntef. And this just shows the pathway. It's pretty big, 130 kb, and here's some metabolite mass spec data and inducible expression. In this case, uninduced worked about as good as inducer one, and from the other side and both sides, obviously it represses it. So this is just a tool for the discovery phase. Once we get expression, we go in and move those promoters around, drop them on the inside at strategic places to turn it on and generate even more product. Here's another project we're working on with some collaborators at Auburn. They had this bacillus pathway. This is a published paper now, bacillucin A, that was very nice antibiotic against MRSA, and we cloned and expressed that pathway. Here's some control results showing our dual inducible promoter, and here you can see the xylose promoter, and bacillus turns on this small molecule lactone-like pathway, and the metabolite clearly is giving nice zones of inhibition with the pathogen on this plate. We're now turning our attention to some biocontrol compounds for oomycete and fungal crop pathogens. And these are examples of soybean being overrun by oomycetes, or cotton seedlings being overrun by fungal pathogens. And there are certain bacillus that produce antimicrobial compounds that hold them in check, as you can see here, for example. And so we're going to start cloning and expressing those here pretty shortly. So just to summarize, here's some of the compounds that I can talk about that we've cloned and expressed using our technology. I've mentioned a few of these, diffocidin and macrolactin and erythromycin are a few of the newer ones, a few of the ones that I haven't mentioned. And we've recently turned our attention to trying to do synthetic biology of natural products from fungi, which are significantly more complicated, 40-ish kb, 40-ish megabase genome, so about 4 to 5 times larger and therefore harder than our biggest streptomyces. And we are pretty sure we're the only ones that can do this precision pathway cloning and expression capabilities. And this is in collaboration with University of Wisconsin, Nancy Keller, and again, Northwestern University, Neil Keller. And we just recently cloned 3 clusters. And I'm just going to show you a little bit of data showing that we think we've got the imizaquin pathway cloned and expressed. And this is some of the data here. Here's a aspergillus with no vector in it. Here's an aspergillus with the imizaquin pathway in it, but no promoter. And here it is with the promoter, and we're obviously turning it on. And we're pretty sure we're getting the right compounds. Still have a little bit more work to prove it out conclusively. And we also have, in conjunction with Northwestern, this AI platform for fungal metabolite production and discovery. You can go to this website and see 1,000 fungi and their genomes dissected according to the pathway and some of the mass spec products that they make. Very nice way to figure out what's going on from a genomic perspective. So that was the genome mining part of the talk. I'm going to quickly tell you about our metagenome mining capabilities. It's another way to access novel drug discovery without having to ever cultivate the bacteria. So I've kind of skipped over some of the parts of this. We've published this. I stuck this in the wrong place. So we've made a large insert library and screened it, and we found some novel chloramphenicol derivatives, and we found a lot of new biosynthetic gene potential capabilities in these 2 publications. And I'll tell you a little bit more about this library. This is the smaller one, 19,000 clones that were made from a soil sample in Georgia, and we've found a lot of new pathways, over 300 small molecule-like pathways, and very low rediscovery rate, less than 0.01%. That's a huge difference compared to what's going on with the classical fermentation screening methods. And the reason why our strategy is going to be more successful than a lot of folks is we use a back vector instead of a phosmid vector. Phosmids only hold 40 kb. In this green zone here, you can see that, whereas backs can hold up to 300 kb. We haven't cloned anything that big from the dirt yet, but we've certainly captured some pretty large inserts. You can see some anti-smash annotated biosynthetic gene pathways here in the blue and the yellow, and we're obviously capturing a lot more of the contents of these unculturable bacteria, and these are actual back clones that have bioactivity. So there's a lot of unexplored dark matter that we're trying to wade through with this process. Going back to the publication, we found out of those 19,000 clones with large inserts, 1,500 of them that contain pathways. So obviously, you know, a big needle in a haystack proposition. This was done by next-gen sequencing using a three-dimensional pooling strategy. And we found a lot of PKS and NERPs and other types of drug-like pathways in there. And we also found some new novel chloramphenicol derivatives. And this is a little bit of a complicated story, but it just shows you there are other ways of working with metagenomic samples. We screened this 19,000 clone metagenomic library in E. coli, and you would not predict that you'd find any new compounds with that approach because E. coli does not support the metabolism of complex NERPs and PKS-like pathways. But nevertheless, we found new chloramphenicol derivatives. And what's going on here is the antibiotic marker, that's the selectable marker on the back vector, is chloramphenicol. And it turns out in some of the clones that we screened for new activities, the chloramphenicol was being rearranged by enzymes on that clone pathway that we had. And they were being remade, rescaffolded, if you will, into some novel compounds. Two of these are new compounds, so it is possible to develop new chemical entities with this biotransformation concept. This is what I should have showed way at the beginning. This is the whole process of making the library, screening it by sequencing, taking the clones that look like they could be metabolite-like drugs, PKS and our nerve pathways, and then shuttling them over to Streptomyces silicolor and then screening them using antibiosis types of antibiotic screens. And we just looked at 3 different bacteria, MRSA, Acinetobacter baumannii, and E. coli, and we found 46 active clones, which is very high hit rate. We suspect a lot of these other clones will have active metabolites, just not in this screen. This shows one of our most active extracts against Acinetobacter baumannii, and we're getting close to isolating it so we can figure out what the structure is, but there's many, many more candidates in our collection that we have to try to wade through. So just to try to summarize here, we've developed some nice novel tools, both genomic and metagenomic, for discovering small molecule pathways, capturing them out of the haystack of the rest of the genome, enriching them, expressing them, and then trying to find out what they provided in the way of structures and activities. And I think that's it. Yeah, so sorry about not getting my slide order correct here, but hopefully it was still pretty obvious what's going on. And you can contact me with this information here. Thank you.
Thank you so much, Dr. Mead. It was really an insightful, informative presentation on drug discovery, microbiome, and CRISPR-Cas system. Thank you so much. If anybody has any questions, please do put them in the chat box for Dr. Mead, and he's also provided his contact, so please feel free to email him for any questions. Will. Thank you so much once again, Dr. Mead.